Today, we're going to start with a quick review of the nervous system. We need to remember these items in the nervous system and the words that go along with the different actions so that you can understand how drugs work in the nervous system. This is how a majority of our drugs will work in the nervous system. Key terms for today would be the autonomic nervous system, the parasympathetic nervous system, sympathetic nervous system, acetylcholine, adrenergic, catecholamine, cholinergic, ganglionic synapse, muscarinic receptors, and nicotinic receptors. And honestly, as I read through those terms, your brain should have been firing off those neurons as you remember what those different things mean. The nervous system is the body's primary communication and control center. It functions in harmony with the endocrine system to allow an animal to respond and adapt to its environment and to maintain a relatively constant internal environment, or homeostasis, through the control of many internal organ systems. The nervous system serves three functions: sensory, integrative or analysis, and motor, which is action. It senses the changes within the environment and within the body, interprets the information, and then responds to the interpretation by bringing about an appropriate action. The nervous system carries out this complex activity very rapidly by sending electric-like messages over a network of fibers. The endocrine system works much more slowly by sending chemical messengers, or hormones, through the bloodstream to target structures. The two systems are very closely interrelated functionally and anatomically. So a nerve will go along with an endocrine gland. The nervous system exerts control over the endocrine system through the influence of the hypothalamus, which is in the brain, and on the pituitary gland, which is also in the brain just below the hypothalamus. The nervous system has two main divisions: the central nervous system, or CNS, and the peripheral nervous system, or PNS, as well as related subdivisions. So some subdivisions of the CNS is composed of what and what? Okay, the brain and the spinal cord. They serve as the control center of the entire nervous system. Get my pen out here so we can do some writing here in a second. All sensory information must be relayed to the CNS before it can be interpreted and acted on. So it needs to go through the spinal cord, through the nerves, to the spinal cord where it's modulated, and up to the CNS where it's perceived. Most impulses that stimulate glands to act and muscles to contract originate in the CNS, so either the brain or the spinal cord. The nerve processes that connect the CNS with the various glands, muscles, and receptors in the body make up the peripheral nervous system. Functionally, the peripheral nervous system is divided into afferent and efferent portions. So remember what afferent and efferent mean. Afferent means that it's going to the nervous system from the periphery of the body. Those are sensory nerves. It's composed of nerve cells that carry information from receptors in the periphery of the body into the CNS. Efferent exits the CNS. It consists of nerve cells that carry impulses from the CNS to the muscles and glands. Anatomically, the peripheral nervous system is composed of afferent nerves, sensory nerves, and efferent nerves, motor nerves, sensory and motor. So here we have the brain and the spinal cord in the central nervous system. In the spinal cord, we have spinal nerves. The spinal nerves send information back to the spinal cord, and the spinal cord sends information through the spinal nerves. The brain, same thing, but these are cranial nerves 1 through 12, and you should remember what those are. So here's the motor system and the sensory system. The sensory system is going to feed back to both the cranial nerves and the spinal nerves. The spinal nerves are going to feed the motor system, okay? And the cranial nerves are going to feed the motor system. The motor system has feedback from the autonomic nervous system. And the sensory system feeds the nervous system. So it tells the afferent nerves um, are going to feed back to the somatic nervous system and get information to skeletal muscles. Whereas the parasympathetic nervous system and the sympathetic nervous system are going to be feeding both muscles, just in terms of blood vessels, but also organs and glands. Okay, so the arrows in this chart indicate the direction of the impulse, so either sensory or afferent or motor efferent. The peripheral nervous system is also subdivided, and I just told you what this is, into the what system and what nervous system. So parasympathetic and sympathetic nervous system in the autonomic nervous system. Okay, or it could be somatic and autonomic nervous system as well. The nervous system that consists of efferent nerves that carry impulses from the CNS to skeletal muscle tissue is the somatic nervous system. It's under conscious control, so it's called voluntary. The autonomic nervous system, whenever I see autonomic, I think automatic because it's involuntary. It's something that your body will automatically do without your thoughtful input. So the ANS consists of efferent nerve cells that carry information from the CNS to the cardiac muscle, glands, and smooth muscle. It's under unconscious control or automatic control, and it's called the autonomic nervous system. The autonomic nervous system has two subdivisions, the sympathetic nervous system and the parasympathetic nervous system. Most tissues innervated, and we'll go back to here so you can see those words, parasympathetic and sympathetic. Most tissues innervated by the autonomic nervous system receive both sympathetic and parasympathetic fibers. In general, one division stimulates an activity by a receptor, and the other inhibits the activity to serve as a method of checks and balances. So we're going to have something stimulate, something inhibit, and we're going to keep the scales even. The fundamental unit of all branches and divisions of the nervous system is the nerve cell. We call that a neuron. Neurons have the amazing ability to transmit information from point to point. The second point may be close or far away, so nearby or at a great distance. It's the length of the axon of that neuron that determines where it synapses. Similar to all cells in the body, neurons have a nucleus surrounded by cytoplasm, but they're different because they have these cellular extensions or processes. These are called axons and dendrites. So the dendrites receive information and the axons transmit information. Axons carry electric-like messages away from the nerve cell. So these go away from the nerve cell. Dendrites carry electric-like messages to the nerve cell. Okay, so dendrites are here, go to the nerve cell, and axons are here, away from the nerve cell. These little branches here are called telodendra. So these are the telephone, okay, telodendra, and these are the synaptic end bulbs. Transmission of these messages along the nerve fibers occur through a wave of charge reversal that moves down the fiber. So there's a wave of polarization, depolarization, and repolarization that transmit this information down the axon. The resting potential fiber has positive charges lined up on the outside of its membrane. Here's a membrane. Here are the charges lined up out here, waiting for the channel to open up so they can go in. Okay, there's negative charges inside the membrane. We have to have a stimulus. Remember, we have our sodium-potassium pump here, taking these charges, 3 sodium, for every 2 potassium. And we've got to shut that off in order to open up the channel for sodium to rush into the cell. Okay? So the stimulus needs to be a sufficient magnitude to that reaches the fiber. And then we have a charge reversal, depolarization, to get the positive inside the cell, which pushes the negative outside the cell. Okay? And this happens in a wave down the membrane. Repolarization is when the charges move back to their original position. So we have the cell ready to go. They're resting, but it's resting, but it's charged and ready to go. And then this depolarization is when the charge is sent out. Axons can be short or long, up to 4 feet in humans, and they end or terminate in as many as 10,000 nerve endings. The large number of nerve endings allows for a great variety in the number and type of connections made with other neurons. The synaptic end bulbs of the telodendra, which are those nerve endings, pass the nerve impulses to an adjacent structure. This is another neuron, a gland, or a muscle by emitting a chemical messenger. So these little chemical messengers are kept in the end of those little bulbs, synaptic bulbs, and the electricity opens up those vesicles and releases that into the synapse. So the messenger is called a neurotransmitter, and it's released into the junction called a synapse that's between the nerve ending and the adjacent structure. So we go from electricity to chemical right here. And then we hit the receptor on the other side, the dendrite on the other side, and we go from chemical, and this is our stimulus to open up the channel to make another electrical wave. So the neurotransmitters, when they combine with receptors on the dendritic side of the synapse, are either going to cause a stimulatory or inhibitory effect. Dendrites may respond to neurotransmitters by generating a nerve impulse, which is conducted via the axon to the adjacent structure, either a neuron, a gland, or a muscle. Neurotransmitters can be either inhibited or activated by the use of appropriate drugs. So we're taking these neurotransmitters that normally occur in the body and we're fiddling with them a little bit with drugs. And we're using our own nervous system with medication to affect what is happening in the body. So here is number one. Here's a normal neurotransmitter. Okay, it's acting normally, getting into the receptor and causing an effect, either stimulatory or inhibitory. This is a neurotransmitter blockade. So this looks like a neurotransmitter, but it's got a little attachment on the end that keeps this receptor from sending a message. So it's going to change. It's either going to inhibit or stimulate. It's going to change the way things work. It's probably going to inhibit with a blockade. Number 3 is mimicking a neurotransmitter. It's a little different. It's the best we can do, but it mimics it, so it may cause the same or a similar effect as a normal neurotransmitter. So we need to keep in mind these different things that could happen at this receptor, because if we talk about this is a beta-2 blocker, then we have to think what a beta-2 receptor would actually normally cause in the body and realize that a blocker is going to do the opposite of that or keep that from happening. Nerve fibers or nerves may have a large diameter. These are called A fibers. A medium diameter, these are called B fibers, or a small diameter, these are called C fibers. Fibers with large diameters conduct nerve impulses faster than those with small diameters. So the bigger the axon, the faster it goes. And even more than that, fibers that are surrounded by insulating substance called myelin transmit impulses faster than non-myelinated fibers. So if you remember that myelin is in little sections that are made up by Schwann cells or glial cells, depending on if it's in the CNS or the PNS. Schwann cells are outside the CNS, right, because of that Schwann truck, that's the delivery truck. And those cells have little nodes, the separations between the cells, where the impulse will jump and leapfrog, and those are the nodes of Ranvier, so that makes it faster. A and B fibers are myelinated fibers, so the bigger it is, the more likely it is to be myelinated, and the faster it goes. Here is an example of, or drawing of, the spinal cord with our dorsal nerve root and our ventral nerve root that come out of the spinal cord and are joined together as a spinal nerve coming out between the vertebrae. So the dorsal root is sensory, the ventral root is motor. And there's a ganglion right here, so there's a connection or a synapse right there. Okay, so we have this neuron, we have some association neurons, connecting neurons between the sensory and the motor. So the sensory is going to sense something and the motor is going to move something. So this most basic impulse conduction system through the nervous system is the motor reflex, or the spinal reflex, happens at the spinal cord. This is composed by a receptor, a sensory neuron, and a center in the CNS for, this is the CNS, right? CNS right here, spinal cord, it's part of the CNS. Here's the PNS, okay? So it's right, that connection is right there, and there's a center for a synapse. And then we have our motor neuron and our effector right here. A spinal reflex can occur even if the spinal cord is completely severed. So if it is severed, if we have it severed, but it's severed like this, we still have that connection there. So if a hemostat is applied to the toe of a dog with a severed cord, they can still withdraw their leg by means of the spinal reflex. What that tells us is that if it is severed, it's not breaking that line there. We still have some connection. Areas of the brain that have importance to the understanding of pharmacology of the CNS. So now we're getting into how the nervous system interacts with pharmacology. Thalamus, which connects everything inputting into the brain with different parts of the brain. It is the operator of the brain. Here's the hypothalamus, which connects and controls the pituitary, right there. The midbrain, which has a lot of autonomic or automatic functions that it feeds. The reticular formation, right here, which controls some pretty important stuff in the spinal cord. The cerebrum is responsible for higher functions of the brain, such as learning, memory, and interpretation of sensory input, vision, pain recognition, and so forth. The thalamus serves as a relay center for sensory inputs from the spinal cord, brainstem, and cerebellum to the cerebrum, and it's involved in pain interpretation. The hypothalamus serves as the primary mediator. I'm just going to go arrow up to thalamus, hypothalamus, just because it's below the thalamus. Serves as the primary mediator between the nervous system and the endocrine system through its control of the pituitary gland. It also controls and regulates the autonomic nervous system. What carries both sensory and motor impulses between the spinal cord and the brain? The midbrain, or the brainstem. The brainstem. Okay. So I'm writing midbrain here, but it's really brainstem that connects to the midbrain. Brain stem. Okay. It contains centers that control vital physiologic activities such as breathing. That's kind of important. Heartbeat, that's really important. Blood pressure, that's important. Vomiting, swallowing, coughing, blood temperature, hunger, thirst, and others. So that's pretty important center for us. If this is broken at all, injury there, the animal will die. The reticular formation is a network of nerve cells scattered through bundles of fiber that begin in the medulla and extend upward through the brainstem. It is a cross that off. It is a part of the the reticular activating center, reticular activating. I'm going to do reticular activating center is part of that reticular formation, and it functions to arouse the cerebral cortex and is responsible for consciousness, sleep, and wakefulness. So if we inhibit the reticular activating center, what is going to happen? We're going to be unconscious, go to sleep, and not be awake. Nerve activity is usually described as the generation of nerve impulses that occur in a dendrite or cell body and then travel down an axon by that electric-like activity, the passage of electrolytes, which is similar to the passage of an electric current down a wire. When this current reaches a synapse or a chemical bridge or neurotransmitter allows that message to be passed to one or as many as thousands of other neurons. These other neurons then carry the message to an interpretation center or a structure that takes appropriate action. So that receptor is important. Neurotransmitters are acetylcholine, norepinephrine, dopamine, serotonin, gamma-aminobutyric acid. Epinephrine is one as well, norepinephrine, epinephrine. The CNS drugs that we use act by mimicking or blocking the effects of neurotransmitters. The autonomic nervous system is that portion of the body, portion of the nervous system that controls involuntary body activities or automatic body activities. ANS fibers innervate smooth muscle, heart muscle, salivary glands, and other viscera. This system operates to inhibit and stimulate to control visceral functions such as gastrointestinal GI motility, rate and force of the heartbeat, secretion by glands, size of the pupils, and various other involuntary functions and characteristics. In contrast to the somatic nervous system, the ANS has two subdivisions. So the somatic nervous system is super easy. It's the afferent system, controls the muscle, skeletal muscle. That's the somatic nervous system. The autonomic nervous system, a little bit more complicated. We have the sympathetic nervous system, which is cholinergic. We have the parasympathetic nervous system, which is cholinergic. Okay, cholinergic acetylcholine. Relate those two things together as parasympathetic. Acetylcholine does work in the sympathetic nervous system as well, but it's primarily found in the parasympathetic nervous system. So cholinergic is para, and adrenergic or adrenaline is sympathetic, fight or flight. Okay, sympathetic nervous system regulates the energy expending activities, fight or flight responses, and the parasympathetic division regulates energy conserving activities, rest and digest. So again, we see this diagram of the autonomic nervous system. It has two neurons that carry impulses to the target structures. In both the parasympathetic and sympathetic side, there are two neurons that go from the spinal nerves or cranial nerves to the affected nervous system. Okay, and where they synapse is based on which part of the nervous system we are. The cell body of the neuron arises first in the CNS, so in the brain or the spinal cord, and then it goes to either a ganglion for the sympathetic nervous system or directly to the target organ where it synapses with a nerve there. The parasympathetic nervous system is craniosacral. So this is where the parasympathetic nervous system is. The sympathetic nervous system is thoracolumbar. So this is the sympathetic nervous system. The sympathetic nervous system has a short preganglionic neuron that attaches to this ganglionic chain first, synapses there first, and then goes to its target organ. And you can see all these target tissues here, and we can understand that the sympathetic nervous system controls items that the parasympathetic system also controls. Okay, and that's just how we get our balance of different things happening within the body. The axon of the first neuron leaves the CNS and travels to a ganglion where it synapses with dendrites of the second neuron. The second neuron then travels to the target structure, and the axons of the first neuron are called preganglionic, and those of the second neuron are called postganglionic. So preganglionic, postganglionic. And then where they synapse at the autonomic ganglion is called the ganglionic synapse. Preganglionic fibers of the sympathetic nervous system are short. They end in ganglia adjacent to the spinal cord. Just picture that pictogram in your mind. The only exception is the preganglionic fiber to the adrenal medulla. The postganglionic, because it actually goes very fast to the adrenal medulla, that is where some pretty important hormones are for this system, the sympathetic nervous system. The postganglionic sympathetic fibers are very long. And the parasympathetic nervous system, it's opposite. So their preganglionic are long in general. They travel to the ganglia located in the wall of the target organ, and postganglionic fibers are consequently very short. So this first here has a ganglionic chain, so we know this is the sympathetic nervous system. The second one goes to the effector organ, and we know this is the parasympathetic. Okay, so we can see effector organs are essentially the same. So normally the target sites of the ANS have both sympathetic and parasympathetic innervation. That's how we get our balance. The physiologic functions of the two systems usually oppose each other and thereby bring about that state of balance. When the balance is disrupted, drug therapy may be indicated to restore the balance. The adrenal medulla, sweat glands, and hair follicles have only sympathetic nerve fibers. Stimulation of the sympathetic nervous system causes an increase in heart rate and respiratory rate and a decrease in GI activity. Dilation of the pupils, dilation of blood vessels in constriction of blood vessels in smooth muscle, and dilation of blood vessels in skeletal muscle. You should be able to fill in all of those blanks just knowing with the sympathetic nervous system what fight or flight is. It's going to cause dilation of the bronchioles and an increase in blood glucose level, so we have the energy to fight or flee. On the other hand, stimulation of the parasympathetic nervous system causes a decrease in heart rate and respiratory rate, an increase in GI activity, and a constriction of the pupils and constriction of the bronchioles. So we can make a drug that acts like the sympathetic nervous system, or acts on the neurotransmitters of the sympathetic nervous system, or acts on the neurotransmitters of the parasympathetic nervous system. Receptors of the sympathetic nervous system are subdivided as follows. We have alpha 1, alpha 2, beta 1, beta 2, and dopaminergic. And knowing what each one of these are responsible for helps you to understand the drugs that we're using. Generally, alpha receptors are stimulatory and beta receptors are inhibitory. The parasympathetic nervous system has the following receptors. This is the cholinergic nervous system, has the following receptors: nicotinic and muscarinic. They accept acetylcholine. Effector organs have one or a combination of these receptors. A drug's effect is determined by the number of receptors in the effector and the drug's specificity for the receptor. The neurotransmitter for cholinergic sites is acetylcholine. It combines with both nicotinic and muscarinic receptors. It actually combines with both sympathetic and parasympathetic receptors as well. So cholinergic sites are found in both the sympathetic and parasympathetic nervous systems. And here are the responses. Alpha 1 targets arterioles, urethra, and the eye, causes constriction, increased tone, dilation of the pupil. Alpha 2 causes constriction of the blood vessels in the skeletal muscle. Alpha 1 or beta 1 affects the heart and the kidneys and causes increased rate conduction and contractility, renin release from the kidneys as well. Beta 2 has to do with skeletal blood vessels and bronchioles. It causes dilation of the skeletal muscles and dilation of the bronchioles. Dopaminergic affects the kidneys, heart, and mesenteric blood vessels. It's going to dilate blood vessels of the kidneys, the coronary vessels, and dilate the mesenteric blood vessels. So it is important that you know what each one of these does. One of my tasks for you this week will be to draw a cartoon of each of these characters, alpha 1 character, alpha 2 character, beta 1 character, or beta 2 character, to help you understand what those each one of those does. The adrenergic neurotransmitters are norepinephrine, epinephrine, and dopamine, and these are the ones that affect those receptors. Epinephrine equally stimulates alpha and beta receptors and is therefore a very potent stimulator of the heart and equally powerful inhibitor of bronchioles. It is the norepinephrine is the neurotransmitter at sympathetic postganglionic fibers to sweat glands and the smooth muscle of blood vessels. So this is at muscarinic sites. Nicotinic receptors are found in all autonomic ganglia, in the adrenal medulla, and at the neuromuscular junction of the somatic nervous system. Muscarinic fibers are found at the synapse of postganglionic fibers of the parasympathetic nervous system and at a few of the sympathetic postganglionic fibers. I do want you to go back and go through that. Make sure you can fill in all those blanks. Look it up in your book. Let me know if I have anything that I misspoke, because that's actually really important for you to do, is to catch things and rework them in your brain so that you understand it. Let me know if you have any questions, and we will not go over this in class. You just need to know it.